There are 6 sections to Biology 2. Cells, tissues and organs, organisms in the environment, enzymes, energy from respiration, inheritance, old and new species. Most human cells are like most other animal cells and contain a nucleus, cytoplasm, cell membrane, mitochondria and ribosomes. Plant and algal cells contain all the structures seen in animal cells, as well as a cell wall. Many plant cells also contain chloroplasts and a permanent vacuole filled with sap. Remember that not all plant cells have chloroplasts, and don't confuse chloroplasts and chlorophyll. Practice labelling an animal cell and a plant cell. You need to know the functions of each part of a cell. For example, chloroplasts contain chlorophyll, which absorbs light energy for photosynthesis. Write the functions of the parts on the diagram. A bacterial cell consists of cytoplasm and a membrane surrounded by a cell wall. The genes are not in a distinct nucleus. Yeast is a single-celled organism. Each cell has a nucleus, cytoplasm and a membrane surrounded by a cell wall. You will need to be clear about the similarities and differences between animal, plant, algal, bacterial and yeast cells. Cells may be specialised to carry out a particular function. Examples of specialised cells are fat cells, cone cells from the eye, root hair cells and sperm cells. Dissolved substances and gases such as oxygen move in and out of cells by diffusion. Diffusion is the net movement of particles from an area where they are at a high concentration to an area where they are at a lower concentration. The greater the difference in concentration, the faster the rate of diffusion. Remember, particles diffuse randomly, but the net movement is from a region of high concentration to a region of low concentration. Do not refer to movement along a gradient. This does not tell us whether it is from high to low or the other way. A tissue is a group of cells with similar structure and function. Organs are made of tissues. One organ may contain several types of tissue. Organ systems are groups of organs that perform a particular function. The digestive system in a mammal is an example of a system where substances are exchanged with the environment. Plant organs include stems, roots and leaves. You'll need to learn the sequence for multicellular organisms. Organism, organ systems, organs, tissues and cells. Photosynthesis can be summed up by the equation carbon dioxide plus water using light energy produces glucose plus oxygen. During photosynthesis, light energy is absorbed by chlorophyll in the chloroplasts of the green parts of the plant. It is used to convert carbon dioxide and water into sugar in the form of glucose. Oxygen is released as a byproduct. Leaves are well adapted to allow the maximum amount of photosynthesis to take place. You'll need to learn the word equation for photosynthesis, and you'll need to be able to explain the results of experiments on photosynthesis. The rate of photosynthesis may be limited by shortage of light, low temperature and shortage of carbon dioxide. We can manipulate the levels of light, temperature and carbon dioxide artificially to increase the rate of photosynthesis in food crops. Make sure you can explain limiting factors. Learn to interpret graphs that show the effect of limiting factors on photosynthesis. Plant and algal cells use the soluble glucose they produce during photosynthesis in several different ways. For respiration, to convert into insoluble starch for storage, to produce fats or oils for storage, to produce fats, proteins or cellulose for use in the cells and cell walls. Plants and algal cells need other materials, including nitrate ions, to make the amino acids which make up proteins. Two important points to remember. Plants respire 24 hours a day to release energy. Glucose is soluble in water, but starch is insoluble. Physical factors that may affect the distribution of living organisms include temperature, nutrients, the amount of light, the availability of water, and the availability of oxygen and carbon dioxide. You can get quantitative data on the distribution of organisms in the environment using random sampling with quadrats and sampling along a transect. You can determine the range, mean, median, and mode of your data. Make sure you know the difference between the mean, the sum of the values divided by the number of the values, the median, the middle value of the range, and the mode, the most frequently occurring value. Different methods can be used to collect environmental data. Validity and reproducibility must be considered carefully, as it is difficult to control variables in fieldwork. Sample size is an important factor in both reproducibility and validity of data. Protein molecules are made up of long chains of amino acids. Proteins act as structural components of tissues, as hormones, as antibodies, and as catalysts. Catalysts increase the rate of chemical reactions without changing themselves. Enzymes are biological catalysts. Enzymes are proteins. The amino acid chains are folded to form the active site. Remember that the way an enzyme works depends on the shape of the active site that allows it to bind with the substrate. Enzyme activity is affected by temperature and pH. High temperatures and the wrong pH can affect the shape of the active site of an enzyme and stop it working. Remember, enzymes aren't killed. They're molecules, not living things themselves. Use the term denatured. Digestive enzymes are produced by specialized cells in glands and in the lining of the gut. They work outside of the body cells in the gut itself. Different enzymes catalyze the breakdown of carbohydrates, proteins, and fats into smaller, soluble molecules during digestion. You'll need to learn the different types of digestive enzyme and the end products of the breakdown of your food. Make sure you know where the different digestive enzymes are made. The enzymes of the stomach work best in acid conditions. The enzymes made in the pancreas and the small intestine work best in alkaline conditions. Bile, produced by the liver, neutralizes acid and emulsifies fats. Remember, food is not digested in the liver or the pancreas. Bile is not an enzyme and it does not break down fat molecules. Bile emulsifies fat droplets to increase the surface area, which in turn increases the rate of fat digestion by lipase. Some microorganisms produce enzymes that pass out of the cells and can be used in different ways. Biological detergents may contain proteases and lipases. Proteases, carbohydrases, and isomerases are all used in the food industry. Remember that most enzyme names end in ase, A's. Some enzymes used in industry work at quite high temperatures, so don't be put off if a graph shows an optimum temperature well above 45 degrees Celsius. Enzymes in detergents break down biological stains such as sweat. They work at low temperatures, so use less electricity, which is cheaper and environmentally friendly. They originally caused problems with allergies, but this has been solved now. The lower temperature washes are less good at killing pathogens, but higher temperatures can denature the enzymes. Enzymes can be produced industrially, both to diagnose and to treat disease. Aerobic respiration involves chemical reactions that use oxygen and sugar and release energy. The reaction is summed up as glucose plus oxygen goes to carbon dioxide plus water plus energy. Most of the reactions in aerobic respiration take place inside the mitochondria. The energy released during respiration is used to build large molecules from smaller ones and allows muscles to contract. In mammals and birds, it enables them to maintain a steady body temperature. Make sure you know the word equation for aerobic respiration. Remember that aerobic respiration takes place in the mitochondria. The energy that is released during respiration is used to enable muscles to contract. When you use your muscles, you need more glucose and oxygen and produce more carbon dioxide. Body responses to exercise include an increase in heart rate, in breathing rate, and in depth of breathing. Glycogen stores in the muscle are converted to glucose for cellular respiration. The blood flow to the muscles increases. These act to increase the supply of glucose and oxygen to the muscle and remove more carbon dioxide. Be clear about the difference between the rate and the depth of breathing. Be clear about the difference between the breathing rate and the rate of respiration. If muscles work hard for a long time, they become fatigued and don't contract efficiently. If they don't get enough oxygen, they will respire anaerobically. Anaerobic respiration is respiration without oxygen. Glucose is incompletely broken down to form lactic acid. The anaerobic breakdown of glucose releases less energy than aerobic respiration. After exercise, oxygen is still needed to break down the lactic acid, which has built up. The amount of oxygen needed is known as the oxygen debt. In body cells, chromosomes are found in pairs. Body cells divide by mitosis to produce more identical cells for growth, repair and replacement, or in some cases asexual reproduction. Most types of animal cell differentiate at an early stage of development. Many plant cells can differentiate throughout their life. Remember, cells produced by mitosis are genetically identical. Cells in the reproductive organs divide by meiosis to form the gametes, or sex cells. Body cells have 2 sets of chromosomes. Gametes have only 1 set. In meiosis, the genetic material is copied, and then the cell divides twice to form 4 gametes, each with a single set of chromosomes. Sexual reproduction gives rise to variety because genetic information from 2 parents is combined. Learn to spell mitosis and meiosis. Remember their meanings. Mitosis, making identical to. Meiosis, making eggs, and of course sperm. Embryonic stem cells from human embryos and adult stem cells from adult bone marrow can be made to differentiate into many different types of cell. Stem cells have the potential to treat previously incurable conditions. We may be able to grow nerve cells or whole new organs for people who need them. Gregor Mendel was the first person to suggest separately inherited factors, which we now call genes. Chromosomes are made up of large molecules of DNA. A gene is a small section of DNA that codes for a particular combination of amino acids which make a specific protein. Everyone, except identical twins, has unique DNA that can be used to identify them using DNA fingerprinting. In human body cells, the sex chromosomes determine whether you are female, XX, or male, XY. Some features are controlled by a single gene. Genes can have different forms called alleles. Some alleles are dominant and some are recessive. We can construct genetic diagrams to predict characteristics. Some disorders are inherited. Polydactyly is caused by a dominant allele of a gene and can be inherited from only one parent. Cystic fibrosis is caused by a recessive allele of a gene and so must be inherited from both parents. You can use genetic diagrams to predict how genetic disorders might be inherited. You can construct genetic diagrams to predict the inheritance of genetic disease. It is important that people make informed judgments about the use of embryonic stem cells in medical research and treatment. There are a number of economic, social and ethical issues surrounding the screening of embryos. Fossils are the remains of organisms from many years ago that are found in rocks. Fossils may be formed in different ways. Fossils give us information about organisms that lived millions of years ago. It is very difficult for scientists to know exactly how life on Earth began because there is little evidence that is valid. We can learn from fossils how much or how little organisms have changed as life has developed on Earth. Extinction may be caused by new predators, new diseases, or new, more successful competitors. Always mention a change when you suggest reasons for extinction. Extinction can be caused by environmental change over geological time. Mass extinctions may be caused by single catastrophic events such as volcanoes or asteroid strikes. Remember that the timescales in forming new species and mass extinctions are huge. Try to develop an understanding of time in millions and billions of years. New species arise when two populations become isolated. Populations become isolated when they are separated geographically, for example on islands. There are natural cycles linked to environmental change when species form and when species die out. In an isolated population, alleles are selected that increase successful breeding in the new environment. Speciation takes place when an isolated population becomes so different from the original population that successful interbreeding can no longer take place.